Sure.
All right. Well, good afternoon, everyone. I'm Sue Kane. I'm going to go ahead and get us kicked off. Welcome to the final Science in Our Valley seminar of the spring series. As you may be aware, this series has been going on for the last 3 years as a way to encourage the scientists of all levels in our community to connect and grow together as a community. And we learn about each other's work, ask questions, we grow our curiosity if you're just joining us for the first time. But when we get all together, we also work on building relationships, and that hopefully takes our work to brand new directions and new collaborations. I've seen some students get some brand new jobs from some of those relationships. So this series is really important, and it will resume again in the fall. In fact, the last 3 speakers who were planned to speak this spring, 2 of those 3 have already rescheduled for part of the fall series, so you won't miss them. They'll be back. And I'll be looking forward to seeing all of you back together again, hopefully with some of those awesome treats that we all enjoy so much. But for today, we're going to connect virtually. And to make that go as smoothly as possible, I've already muted you. I'm going to ask you to stay on mute until the end of the presentation. If you have a question that pops up along the way, please feel free to add that in the chat, and we'll circle back to those at the end of the presentation. If you're a K-12 educator and you're going to be seeking clock hours for participating today, please include your name in the chat as a virtual kind of sign-in. And if you would prefer, you can also private message me, Sue Kane, that name, and I can record it for you. With that, I'm going to introduce our esteemed speaker today. Dr. Jessica Waite is a graduate, or is a research geneticist and a molecular biologist. She'll be joining the USDA Tree Fruit Research Lab, but she's been in Dr. Calcett's lab at WSU. Before that, she was a Husky, and so did her bachelor's degree and PhD at University of Washington with areas of specialty in plant biology and development. And she spent some time at the research labs at UW and Fred Hutch, and of course WSU, and now headed to the USDA at some point here soon. She works in understanding the complex relationship between molecular mechanisms and hormonal signals as they impact growth and development in plants. And so we're really lucky to have this opportunity to dive into her work this afternoon and learn a little bit more about these molecular systems that help acclimate apples to heat stress. And with that, I am going to turn it over to Jessica to share her screen with you.
Right, thank you. Here, let's get this. Okay. Can everybody hear me? It's all good. Okay, great. Well, thank you so much to Sue and Soon Lee for organizing and inviting me to talk today. And thanks for coming to this last Zoom seminar. I was surprised I was the last one of the group, but it makes sense. Everybody's moving to fall. And as Sue shared with you, I am delighted to say that I've just started as a research geneticist remotely for now, of course, at the USDA, which is across the parking lot from where I was working in Lee Calcett's lab. And there I'll be working on pear genetics and genomics, focusing a lot on pear rootstocks. But today, I wanted to share with you some of the findings and some of the project plans and research I've been doing in Lee Calcett's lab as a postdoc at WSU Tree Frec, and what we found over the last couple years. As I don't really have to tell any of you, I think everyone in the room is pretty familiar with the fact that apple growing regions in Washington State experience really intense levels of heat and light and drought stress. So for this map, what we're seeing here is the amount of rainfall per year. And you can see where, can you guys see my mouse? Is that showing up? Okay, awesome. So all of this sort of central region where so many apples are grown receives some of the smallest amount of rainfall over the year. And as you've experienced the air temperature yourselves, the apple fruit with the high amount of sun, the low amount of cloud coverage, the high heat, What this does to the surface of the apple is it starts to really heat up this tissue. So the fruit surface temperature can get up to 20 to 30 degrees Fahrenheit higher than the air temperature during some of the warmest parts of the day when the sun is shining directly on the apples. And for the rest of the talk, I'll talk a lot about FST, which I mean fruit surface temperature, and that will keep coming up. When fruit surface temperature goes above a certain temperature, it will start to lead to heat-dependent sunburn developing, which I'll talk a little bit more in a second. And just to show you an example of the kind of heat stress that apples are experiencing in the light, this is a graph of over the summer of 2018 of the daily predicted maximum fruit surface temperatures, and that is a prediction that comes from air temperature, fruit size, how much wind is in the air, sort of how much direct heat those apples are getting. And what's marked on this graph here are sort of the below, at, and above ranges of fruit surface temperature that lead to sunburn. So you can see in 2018, there were quite a few days where the many apples experienced this above sunburn pressure, threshold temperature pressure. So when I'm talking about sunburn, for the rest of this talk at least, I'm really talking about the sort of pre-harvest sunburn, the physiological skin disorder that's happening pre-harvest. There's also a lot of implications for how this affects post-harvest sun scald, and a lot of folks around here are working on that. But I'm going to focus a lot more on what's happening when the apples are on the tree. So there have been a few different kinds of sunburn described, largely by Larry Schrader, who was here at WSU before. And so one of these kinds is sunburn necrosis. And sunburn necrosis, I forgot to fill in temps here. Sunburn necrosis occurs at a high heat threshold. I believe it's about 50 degrees and above Celsius, about 125 degrees Fahrenheit. And once fruit reaches that temperature, it is going to start losing the tissue viability, like the tissue is going to start dying. And this is regardless of whether light has shown on it. Even if they were reaching that temperature in a box, they would still start to develop these symptoms. Sunburn browning happens with high heat, but less than that necrosis temperature. So this is about 113 to 125 Fahrenheit, and leads to a discoloration of the fruit, and this requires light. So if you have a sticker covering it or something else in the way, you won't actually get this browning effect. And then photooxidative sunburn is kind of a light shock. So this will often happen if a fruit is shaded and then and not sort of gotten used to the light in the environment, and then you quickly move it out of the shade, you'll get this bleaching effect. And what happens to the fruit tissue? There's also a nice graphic from a review that Larry Schrader put together, which kind of shows if you just increase temperature, you can get sunburn necrosis. If you have light and temperature, sunburn browning, but then if you get past that threshold, you start to develop necrosis. And photooxidative sunburn, you can also get necrosis from if you increase temperature or increase more direct light. And like I said, necrosis leads to tissue death and loss of membrane integrity, whereas sunburn browning, you mostly get discoloration, and then this can lead to a lot of post-harvest problems as well. But at this point, it is sublethal. And then photooxidative sunburn, you get a lot of this reactive oxygen species production, and you get this bleaching and potential eventual necrosis. So what can we do about this? Currently, there are a number of different methods to mitigate sunburn. So one of these is evaporative cooling. So this is where sprinkler systems are turned on at a certain temperature. Currently it's 85 degrees of air temperature. And then the water settling on the fruit and then evaporating is able to cool the fruit surface temperature. Overhead netting is starting to be employed in a lot of places, which keep the fruit shaded to lower the fruit temperature. And then protectant sprays, essentially sunscreen, in the form of either kaolin clay or there are wax-based formations. These can, well, essentially, like I said, give a sunscreen effect. And all of these have their different drawbacks. The protectant sprays can be really hard to get off. Overhead netting can be pretty expensive. And then using a lot of water in these evaporative cooling systems can lead to some water-based problems, including things like anaerobic soils. And so we are very interested in, and Lee and Samaya, who worked on this project before me, interested in how we can learn more about how and why sunburn happens, so that we can hopefully inform how to use these systems, or whether and how we might be able to use these systems more efficiently. Oh, yeah, so this is pretty much what I said. So can we learn how and why apples get sunburned and how they are able to protect themselves? Learn more about these mechanisms for sensing and responding to stress. And then can we use that information to inform more efficient mitigation strategies and decrease losses to sunburn? And then can we potentially use this information to develop new models or integrate this into models, as well as develop genetic markers for breeding? So at this point, I'll give you a little roadmap of where I want to go for the rest of the talk. So there are two sort of avenues. The first is sunburn, and then I want to talk a little bit about thermotolerance and acclimation. So in talking about sunburn, I just want to give you a bit of background on what we already know about how sunburn happens in apples, including some of the physiology and biochemical studies, and some of the gene expression studies, and then talk to you about what we're hoping to add to this with our studies, as well as sort of introduce this idea of acclimation or acquired thermotolerance, and what we know from model systems about this and crop systems, and then what we have found in our apple experiments as well. So just to give you a little bit of background on what we already know about sunburn, there's been a lot of work early on sort of characterizing what is happening to apple fruit when they experience this very high heat stress. A lot of this work was done by Larry Schrader's lab. So he put together a model for determining the fruit surface temperature. So here's just sort of an example of how with the air temperature, you can see that the fruit surface temperature increases and then decreases through the day. And that is dependent both on fruit size, on the air temperature, but also the direct sunlight, the photosynthetic active radiation. And as I learned for myself this summer, or last summer, the amount of wind that is out in the orchard drastically can lower fruit temperature quite a bit. We had a fairly cold summer last summer. So models are a model built to determine fruit surface temperature was developed. In more recent years, there's been a lot of work looking at sort of targeted analysis of certain groups of genes that might be involved in sunburn, as well as a lot of metabolism and biochemistry. So here I'm showing you some work done to look at heat shock proteins and what they're doing when the apples are going through these different dips or mountains and valleys of temperature, of air temperature. So you can see that some of these heat shock proteins, you see a definite increase through time as you get to hotter points. A lot of work has focused really recently in a number of labs, including some of the work that Carolina does, looking at the phenylpropanoid pathway and things that are leading to things like pigments, like anthocyanins, different flavonoids, looking at how the enzymes involved in making these compounds are active, but also the gene expression of the underlying genes of all of these compounds are coming up and down. So you can see, so what they did in this experiment was they took fruit that had been shaded, and then they turned the fruit, and over this time period, you can see 0, 6, up to 174 hours, they looked at gene expression of all of these genes involved in this pathway over this time period. And you can see after being exposed to light and heat, you get this real jump in gene expression in a lot of these genes. So we have some definite background in sort of what's going on, what pathways are being turned on, what genes are being turned on when apples are exposed to heat and light. And so what part of what we were doing is sort of being able to add to this. Something that I got interested in adding since I've been here is looking at gene expression over time, like they did in that last experiment, but looking at a whole suite of genes over time. But also being able to compare this work to the acclimation work, which I'll tell you about here in a second. So our Sunburn experiments were set up where we treated the fruit in the field. This was early in the season before they had experienced a whole lot of different temperature changes. Treated them with below fruit surface temperature threshold for sunburn. We actually treated them with an treatment as well, and then above. So this is where you should be starting to get close to that upper range of sunburn. And then collected tissue to look at gene expression, to look at pigments, look at biochemistry after 24 hours, 48 hours, and 72 hours. And then at 72 hours, we also looked at all of their sort of physiological measurements. So we looked at pigment or spectrometry, so looking at pigments in the field, as well as position on the tree and size of all the fruit, etc. And so, what was I just gonna say? I can't remember. But to do these experiments, what we did is we actually just bagged the fruit. So I think the first year, Samaya had started by using a magnifying glass method, but it was really difficult to keep in place and hold fruit at a certain temperature. And so what she switched to and what we did the second summer was to just use plastic bags, and you can kind of open the bags when the fruit get too hot and close them up when they're too cool. And then we used, you can see some of them in this picture, thermocouples. So we just kind of plugged these wires into the fruit surface to manage or to monitor temperature. And then we also had infrared sensors to hold up to the fruit to monitor temperature. And so kept them all there for about 20 minutes and then let them develop. Sorry, that's what I was going to say. The symptoms of sunburn take about 72 hours to develop. So that's why we're doing our physiological assays at the end of 72 hours. But we want to catch what's going on in gene expression before this happens. So this is just an example of how one of the things we do at the end, which was to call sunburn severity. So these are two different scales for green fruit. So here's Granny Smith on the bottom and Honeycrisp on the top. And so when you see in some of the graphs I'll show later, we call this this category at the bottom where there's absolutely no sunburn showing, that would be sunburn 0, and then 1, 2, 3, and 4. So that's what you're seeing in the upcoming graphs. So here I'm just going to show you an example of some of the sunburn data that we've collected, because at this point this was largely proof of principle to make sure that this experiment worked, and gather data on the sunburn conditions. So this is data from this last summer, 2019. And you can see, so this is control fruit. And again, this is the beginning of the season, so we don't expect them to have much sunburn at this point. And you can see most of these fruit are 1s and 2s. We did have one hot day right before we started these experiments. So the Granny Smith have a few 3s in there as well. When we treat with that below stimulus, so they're getting a high temperature, but sublethal, we see the shift a little bit. Oh, where'd my mouse go? Where we see the distribution goes a little bit more towards 2 and 3 for the Granny Smith. It's pretty similar for Honeycrisp. But when we treat with this above stimulus, above fruit surface threshold, temperature threshold, then we're starting to see more 2s and 3s and quite a bit more 3s in Granny Smith. So this was good to see that our sunburn treatments were working. We also, so I'm going to show you a little bit of data from 2018. We haven't gotten this data yet for 2019, but this is just looking at some of the pigment accumulation in the apples from those years. And you can see in Honeycrisp, so looking at anthocyanins, which develop in a lot of stress situations, you can see in controls compared to below, at, and above, we're getting more anthocyanin accumulation in these higher treatments. And we're seeing that as well in Granny Smith. We also looked at chlorophyll. So this is showing the chlorophyll AB ratio. And this is something, chlorophyll is known to decrease under sunburn conditions or under sunburn. So we do see this decrease in Honeycrisp with temperature. We did not actually see a big decrease in Granny Smith at this point. But we are seeing that they are responding to these different heat treatments. Something that has been done a little bit in the field, there have definitely been reports on the fact that different cultivars behave differently as far as how well they respond to sunburn conditions, how well they acclimate to sunburn conditions. And so the first summer, one of our undergrad students for this summer went out and collected fruit surface temperature from 2 different cultivars, I think every day or most days of the summer. And that, when you compare that to the temperature and photosynthetic active radiation at the time, so the direct sunlight, you can see that they have differences in fruit surface temperature in response to the same environmental conditions. So what you're seeing here on this upward axis is the fruit surface temperature. And along this edge is the light photosynthetic, oh my goodness, sorry, that is such a hard thing to say. Photosynthetically active radiation, and then air temperature on this axis. And so you can see Granny Smith, the sort of shape of that curve, it looks like that, whereas Honeycrisp actually gets quite a bit hotter for the exact same environmental conditions. So something that I don't have data for yet, but well, I guess let me back up for a second. So something we're interested in looking at is looking at the gene expression dynamics. And we're planning to run an RNA sequencing experiment so we can get sort of a snapshot of what all of the gene expression is doing in response to these these different sunburn treatments or different temperature treatments over time. And so at this point, we're getting that RNA ready to send out, but then Corona happened, so that will be sent out as soon as we can to be able to sort of compare and get a good idea of overall what kinds of genes are turning on in these different conditions. And I'll talk a little bit more about that later. But what we did with the data, or with the samples from the first year, is we took a candidate gene approach. So we chose a number of genes that are involved either with heat shock or with acclimation or with some of these phenylpropanoid pathway genes. And we looked at their gene expression of just these genes in the samples from 2018. And so you can see here, so Honeycrisp is on the top and Granny Smith is on the bottom. And these are just two, so MIB10 is an anthocyanin biosynthesis gene, and LDOX is associated with that same pathway. And while we didn't see in this data where we're just looking at time point 0, or just looking at the 72-hour time point, we didn't see any significant changes in gene expression. We do see these trends where we are getting this increase with these higher temperatures. But what we want to do with the data, or what we want to do with the samples that we collected this last summer, is kind of do this large scale comparison. And so what this graphic is showing is what some of these different comparisons might tell us. So when we can compare the control samples versus these treated with above, we will be able to see which genes are up in control, which genes are down, to get a sense of the genes that are involved in sunburn initiation and development. What I'll talk a little bit about later is what we're hoping to see in the comparisons between control and below. What we're thinking is genes that are coming up in this low heat treatment might tell us something about acclimation, but I'll get to that. And then just being able to compare all of these different time points, we might be able to see dynamics, which genes are coming up early, which genes are coming up late, and what that might be able to tell us about what pathways are turning on. And then I'll talk about the acclimation portion in a second. Oh, actually here. So the second part of the talk I kind of want to focus on, which is a large part of why this, this project got started, was thinking about acclimation to largely heat stress. And so I just want to introduce a little bit about acquired thermotolerance, which so far seems to in the field be pretty interchangeable with the word acclimation, or at least that's how I'm going to use it today. So acquired thermotolerance equals acclimation. So this is the process where an organism experiences sublethal stress. So this is like that below threshold sunburn temperature. And what I'll call this for a lot of us will be the priming stimulus. And that initiates signals, turns on genes, turns on things potentially like hormone signaling, and makes epigenetic changes so that these programs get turned on to increase defenses to protect against future stresses. So that's just shown here in this cartoon, where if plants, for example, are treated with a heat stimulus, you might turn on all these programs. So then when they get treated with this higher heat stimulus that would otherwise kill plants, they're already defended against it. So they will survive that heat stimulus. Whereas if you don't pre-treat and don't get that priming stimulus, you might, when you're faced with that higher heat treatment, then the plant might die. So this is acclimation in a nutshell. So when I first got to the lab, I got really interested in reading all about different kinds of thermotolerance. And there's a bunch of different types of pathways and genes that are turned on in different situations. But they all kind of follow this general trend where you're treating with this sublethal stimulus, and that protects against a otherwise lethal stimulus later. So there's basal thermotolerance. So that's just things that get turned on in response to stress to protect an organism without any acclimation. The short-term acquired thermotolerance, this is, at least in model systems, has been shown to be things that get turned on, programs that get initiated for a short period of time, a matter of hours. To prepare for a high lethal stimulus, whereas long-term acquired thermotolerance might be active for a few days. And this is a big open future question, I think, for the Apple work, is whether acclimation falls under long-term or short-term, as well as how long that long-term is for apples. In Arabidopsis, it's a few days. What is it in sort of plants that have survived or have evolved to be, you know, perennial, out in the field, out in these very intense conditions all the time. And then there is also, I'm not going to talk about it much today, but this thermotolerance to moderately high temperatures. Every time I see this acronym, I think Teenage Mutant Ninja Turtles, but it's an H, not an N. But this is, I'll show you an example of this later, but this is where you're getting sort of low-level heat waves for a long stimulus, and then that will initiate some of these thermotolerance programs. There we go. So a lot of what we know about how this works is from model systems. Examples in rice and Arabidopsis have been able to identify genes that are involved in this long-term, these are both involved in long-term stimulus, and then our long-term acquired thermotolerance, as well as a number of studies in tomato. This one is just an example of actually in programs coming on in response to chilling. And these last for quite a while. And then there's some pretty recent studies, both from 2018 in different non-model systems. So what I'm showing you here is one of those examples that's probably more of the thermotolerance to moderately high temperatures. So these are alpine blueberry leaves. And so what they did is they took these heat hardening chambers out to the field, and they subjected plants to 7 days at 30 degrees C. And that was able to protect them quite a bit from later heat in the season. And they also found that this was dependent on light. So this bottom part of the figure is plants that this was done in the dark. So a lot of these pathways are sort of interconnected between, between light and temperature. There was also a really nice experiment that came out in potato. So this is where they gave the plants a 48-hour heat shock, kind of like our below treatments. And what they found, so these guys on the right were acclimated for 48 hours. These guys on the left were not. And after this severe heat stimulus, these acclimated plants fared much better. They look actually pretty normal. And I apologize for the graininess of the picture. So a big question that Lee and Samaya started this project on, and that I kind of joined into and got really excited about, was, can apples acquire thermotolerance? And so this is the experimental setup that we used, where we treated a whole bunch of apples with this below stimulus first. early on in the season. And then the first run of this was to look at just challenge the apples at the end of the season with below, at, and above fruit surface temperature sunburn thresholds. And then collect and measure these after 72 hours. And again, these are the same situation where we bagged the fruit and treated them with these temperatures. And so these are the sunburn call data results from 2018. And what I'm showing you is this is all of the fruit grouped together. So below, at, and above fruit grouped together, whether they had been primed first or not primed at all. And what you can see here in Honeycrisp is that compared to controls, which we have quite a bit of these higher categories, Sunburn 3 and Sunburn 4, these were drastically reduced in fruit that was primed. And this was true even more so in Granny Smith. So this method really shows a lot of promise and really wanted to repeat it last summer. But what happened last summer was that the temperature just did not cooperate for the experiment. The temperatures were great for apples growing in the field, but we didn't get a lot of pressure, sunburn pressure last summer. And so we weren't able to repeat this part of the experiment. So again, showing you a little bit of some of this data from 2018. So I showed you these graphs before. This was anthocyanins and chlorophyll ratios at all of these different temperatures. So these were, before the fruit or without any priming with that stimulus. When we prime the fruit ahead of time, what we see, so comparing this first upper left graph where we get this relationship with higher temperature and higher anthocyanins, is we lose that relationship. So it seems like anthocyanins are not going up with these temperature increases when fruit are primed. And this was true for anthocyanins for Granny Smith as well, which makes sense because anthocyanins being something that comes up under stress and under sunburn, we might expect to see this, but it was nice to see. And this was also, this was not true of chlorophyll, or at least the chlorophyll AB ratio. These seem to, decrease with temperature and Honeycrisp regardless of priming. So it seems like some of these pigments are very related, some are not to this priming. So I just wanted to, what I was about to say or show you an example of is the differences in temperature between 2018 and 19. So you can see these are hourly temperatures, and I showed you the daily ones at the beginning, but 2018, we see these are air temperatures, and 85 is the recommended time to turn on the sprinklers. So we can see a lot of time spent above that 85 mark in 2018, and 2019 really it was very little in comparison. But coming back to this a little bit, now that I've talked a little bit about acquired thermotolerance, this is why we think comparing this control condition versus the below condition might give us a sense of what genes are coming on sort of from this priming stimulus that might tell us a little bit more about which are involved in acclimation. Because it seems that through all the studies and model systems, there's a suite of genes that comes on when you give that sublethal priming. So just to tell you a little bit about where, besides the RNA-seq experiment where we want to get an idea of the whole snapshot of genes that are coming on in response to all of these temperature conditions. Another thing that I think will be really important moving forward for sunburn work, and particularly for this acclimation work, is trying to understand more about that priming stimulus, why it works, how long it needs to be, how long it needs to last, yeah, how long it needs to be and how long it needs to last, and how early in the season it might be important or how late. And so a couple of experiments that I set up this last summer, but again, was not able to see through because the temperature did not cooperate. But some of these ideas were treating with this below stimulus at different time points before the end of the season or before a certain collection point. And the other one that I would be very interested in, or that I attempted to do, was to use shade netting to get at this question. So treating fruit that have not been exposed at all, that have been netted the entire time with this below stimulus, and then looking after 3 days after, or removing the nets to expose the fruit to sunburn conditions, 3 days, 7 days, and 14 days later. Yeah, and then collecting after 72 hours. So that's something that I think moving forward will be a really big question, is the quality of the priming stimulus. So with that, I just want to thank Lee especially for being able to, have this position and work on this project. Samaya, who did a lot of the foundational work on starting this all. Brenda and Alex have helped a lot in the field. And Alex has been helping a lot with RNA extractions and getting a lot of this prepped to go out for the RNA sequencing experiment. Orlando and Michelle, who's now the lab tech in Lee's lab, helped a lot with measurements, and a number of other folks that have helped at different aspects of this in Lee's lab. And then, yeah, funding from the Washington Tree Fruit Research Commission and Washington State University. And yeah, I can take questions then.
Right.
That's it. I thought it was talking about it. So
Right.
for whatever reason, going through, but
You are able to unmute yourselves now and ask questions. And I will kick you off with a question just to get things rolling, because I was really early on, you were talking about the reactive oxygen species that can sometimes happen. Does that impact your ability to do some of the molecular measurements? Like, do those degrade your DNA or your RNA, I guess it would be, when you're doing some of that molecular analysis?
Yeah, so luckily, I mean, I think there's probably some of that going on, but luckily I think the largest response in reactive oxygen species is in that photooxidative sunburn, which we weren't dealing with as much here. These were fully exposed to sun the whole season. So luckily we didn't have to, yeah, we weren't dealing with as much of that type of sunburn.
Got it.
Thanks so much for your presentation. I had kind of a practical question, and apologies if I missed it. When we think about the damage that's done to fruit through heat and light exposure and the impact on growers and the economy, what's the magnitude of the problem that we're facing so that your research is addressing?
How did I not say that? Some of the studies have estimated that it's up to like 10% of fruit loss due to sunburn related problems. Yeah, it's a pretty significant amount. Yeah, I always have that little bit in there, but forgot to say it.
Hey Jessica, thank you for your presentation. I have a question. Do you happen to know what the thermal capacity of apple fruit is?
The thermal capacity. So I guess that would probably change from cultivar to cultivar. But I guess
So I'm wondering what the deviation or differences are across cultivars or even across developmental stages of apple.
that is a good question.
Okay.
That would be good to figure out. There have been a lot of like different studies on different cultivars, but I don't know that anybody's taken like a really careful look at across a bunch of cultivars. So that's really, I think, what a lot of what that the figure that Brenda worked on, looking at fruit surface temperature over the season. But I don't think it's been done in a whole bunch of cultivars to see sort of the maximum that they can deal with before hitting like necrosis.
Got you.
But that's a good question. Yeah.
Jessica, this is Jim.
Hi.
So I'm wondering about the whole acclimation part of things, and
Mhm.
you know, we live in this climate where, because it's so dry, we have for most of the summer this really amazing day-night temperature differential,
Yeah.
and you know, we have days clearly well above 100 degrees, and it can be in the 60s at night.
Right.
So it seems like that environment is really not very conducive to acclimation because you have so much of a period of the day when the temperature is low.
Mm-hmm.
Does that, and so one of the things I'm thinking about, if that is in fact the case, because it seems like in years where we have hot nights,
Mm-hmm.
there's actually more potential for at least some of these problems, maybe not sunburn per se, but other types of food quality issues. And so, you know, it's not so much a question as it is a comment, but I think the question potentially is, when I think about the sunburn issues that we have in the Pacific Northwest versus what there is in Eastern North America,
Mm-hmm.
where certainly it doesn't get as hot, but it stays hotter at night than it does here.
Right.
And so maybe potentially that's a better environment to be acclimated to heat stress during the day because it doesn't cool off as much as at night.
Yeah,
Does that fit with the whole acclimation scenario as you perceive it?
Yeah, that's a good question. I am definitely very interested in how nighttime temperatures affect some of these things. That was something that we kind of proposed to do. We're really interested in potentially in the future building chambers to have in the field, so we could be able to, you know, control some of those aspects, especially for some of these longer term experiments, because I'm really curious, how nighttime temperatures change the stimulus, as well as sort of duration. What I think also may have been the case, like it may depend more on, or that one aspect of that acclimation stimulus, I think is just that high, or that high temperature in the day and how long it lasts. So I guess where I'm going with this is, when I look at 2018's data, what it looks like is when the treatment was given, that below sublethal treatment, it seemed like a few days after that is when the high temperatures sort of started to peak again. And so what I'm kind of potentially hypothesizing or playing around with is the idea that the stimulus doesn't necessarily last more than a few days or a week, but it kept in that first season getting sort of re-upped throughout the rest of the season. But it may have had to do with nighttime temperatures as well. That is a very good point that I think could have a big effect on it, but there's not as much work on that as far as I can tell.
Jessica, this is Carolina.
Hi.
Hi. I have two things in my mind in regards to sunburn and sunscald too. But first of all, is we've never been able to really understand why a fruit next to each other in the periphery of the tree or the canopy can actually, one can burn bad and the other one might not, or might just. So in my head, this is something to do with the energy that tissue accumulates and translates into functionality or not of the different processes involved in protection of that tissue.
Right.
And so that's one thing. And the other part is circadian rhythm of all these enzymes involved in all different pathways throughout the day, night, and then next morning. And it's complicated, but I know that, you know, you might have some good ideas because of your background to know how to assess that and then this energy issue. So quantification would be something to do on different parts of the apple. There is only one paper I've seen where they show different accumulation of pigments and symptoms changes from the outside of the halo that receives this direct sun to the middle of it. So you know there is energy involved in there. It's not the overall air temperature or tissue temperature. It's actually that change in temperature. It's pretty complex going to that little space of research in the organ. But anyways.
Right. And that's something that, you know, we were trying to control as much as we could for circadian rhythms for this first part, to be able to collect them at the same time of day so that we weren't seeing those effects or weren't being complicated by those effects. Because it definitely seems that, these will change drastically throughout the day, potentially, which actually hooks into the cold nighttime temperatures as well.
Right, absolutely.
Light and everything is all so connected.
Yes. And the energy would be the other thing that you might look. And that is being really creative in the field to assess the amount of energy that fruit gets, one compared to another, you know.
Yeah, because that's something that I'd be really interested in being able to do, is to initially be able to totally control for some of those outside forces, and then be able to add them back in, you know, by using different kinds of chambers to control the entire atmosphere, and then compare that back to field conditions. But it's not easy to do.
No, it's not. And maybe just measuring, you know, different spots of the apple tissue that is exposed, unexposed.
Right. Yeah. Good point.
Hey, Jessica. Going along the same line as of Carolina's question, I'm wondering, so do you see differences where if the fruit is in isolation, it gets more severe sunburn compared to fruits that are sort of being in a cluster with other apples?
I was just thinking about that when she was saying that. Whether they are having to sort of like compete on the same branch, or
even how the light can get diffused in some manners that would help.
Right. Yeah, no, and that was something we did not initially pay attention to, but that is a super good point.
Okay. Or even if you have like a heat vision and just scanning the heat vision over a cluster of fruits and see whether or not are there differences where you see certain hot spots of heat.
Yeah, and that was something like when we were out doing this, we were definitely, like I said, we weren't super paying attention to it, but I will say I'm sure that we focused more towards single fruit because it was easier to get the bags around them. And so for the most part, a lot of our fruit was in that more exposed category so that we could easily manipulate them in the same way.
Okay, thank you.
Mm-hmm. But yeah, that would be something to really, really pay attention to in future experiments.
Hello, Jessica.
Hey.
Good job. And,
Thanks.
yeah, just to confirm, in my recollection, we did tend to go for fruit that were single on the trees.
Mm-hmm.
But something I was wondering, it looked like you did chlorophyll and anthocyanin analysis.
Mm-hmm.
I was wondering if you considered looking into carotenoids as well.
Yeah, so I started doing all of this before like Carolina was here and all set up. And so I was basically just doing that initial analysis on doing some really basic extracts and using a plate reader. And I was not able to find any great protocols to be able to separate out carotenoids from other things. So yeah, I'm definitely interested in including it, but that first year was not able to. It was too hard to do with the limited equipment that we had.
All right, great, thank you.
Mm-hmm.
We probably have time for one more question if there's one more question out there. All right. Well, I think on that note, I'll go ahead and wrap us up. Thank you so much for taking the time. This was awesome. It got my brain thinking in all kinds of different directions. So I'm excited to learn a little bit more about it and excited to see where things go.
All right. Thanks for everybody for coming.
Thanks so much.
Thanks.
Great job, Jess.
Thanks.
Good job, Jessica.